Hello, I'm István Peták, founder and CEO of Oncompass Medicine. I'm a cancer researcher. I'm also a researcher at Semmelweis University, and I'm also an adjunct associate professor at the University of Illinois at Chicago. It is my privilege to be the first author of our paper published in Nature partner journal Precision Oncology. We worked together with Professor Turno's team to test our computational method called digital drug assignment and the computational tool which implement this method on clinical trial data generated in Shiva-01. We took the data about the molecular profile of the 113 patients actually treated with targeted therapies in the trial. We uploaded these results into our computational tool, the real-time oncology treatment calculator, and then we ran the algorithm computed in the software tool to assign a specific mathematical score called aggregated evidence level to each genetic alterations, and also druggable targets linked to these genetic alterations, and also to targeted therapies, molecularly targeted therapies, targeting these druggable targets and linked to these potential driver alterations in these patients. The computer created a prioritized list of alterations and a prioritized list of targets and compounds. What we found was that in patients who had clinical benefit from molecularly targeted agents, the average level of the aggregated evidence levels was 3 times higher than of those which failed to achieve any clinical benefit in the group where patients progressed on therapies. So that was the first sign that there is a link between the predictive mathematical score, the aggregated evidence level, and the outcome, the response to target-based targeted therapies. Then we grouped patients into 3 tiers. One tier representing patients with very high level aggregated evidence levels, and then a low tier where the aggregated evidence level was actually minus, below 0, which were designed to predict resistance to therapies, and the group in between. What we found was that in patients with the high aggregated evidence levels who received therapies with high aggregated evidence level above 1000 actually achieved around 70% disease control rate, while patients who received low drugs with low evidence level actually had only 17% disease control rate. Also, patients with the high AEL level TR had significantly longer progression-free survival compared to those with low aggregated evidence level. These results show that a computational tool which now can take into consideration multiple evidences, hundreds or thousands of evidences, and can take into consideration the whole molecular profile of cancer, can actually improve our decisions when we try to choose the most effective targeted therapy for our patients. So this tool can not only help us to choose the right or the most effective off-label therapy, but we can always optimize our decisions on different lines of therapies when we have to make a decision between registered treatment options, or when we have to make a decision whether to refer a patient to a clinical trial or not. But of course, when we run out of registered treatment options in that tumor type, we can still use this system to to prioritize targeted therapies for our precision oncology practice and to make a confident decision to choose the therapy with the highest evidence level and with an algorithm and a method, a computation method, validated based on clinical trial data. And we are very excited about working together, oncology centers and oncologists all over the world, to make this next step in precision oncology and moving from the era of single parameter companion diagnostics and biomarkers to multi-parameter based precision oncology and into the digital age of precision oncology, when we finally can take into consideration the whole picture of oncology, the whole picture of the genomics of cancer, and to make the next big giant step toward our dream to provide effective therapy for all our patients. Thank you very much for your attention.